not because I'm saying we shouldn't use a vaccine, but that we can improve it and we can make it better and we shouldn't ignore problems. Problems are our teacher.
Welcome to this special episode of Healthcare Unfiltered. I am your host, Shadi Nabhan. I'm a hematologist and a medical oncologist with interest in all aspects of healthcare delivery, treatment, leadership, mentorship, and policy. Today's podcast is with Professor Richard Burt, who wrote a book called Kill Switch. It's a fascinating book. Some elements of the book are rather disturbing, but nonetheless, it's a book that I highly recommend all of you read. The book talks about the history of viruses, and Richard, Professor Burt, starts from early on with smallpox, moving all the way until COVID-19 and the pandemic. Professor Burt talks about these viruses, what they are, how they evolved. Some of these viruses brought down empires. He also talks about vaccines and how these vaccines were developed in order to mitigate and to minimize and sometimes eliminate these viruses. There's a lot of history in this book, a lot of research that Richard did to bring these facts. But towards the end, he focuses on COVID-19 and the pandemic, and he reveals a lot of information some of us probably did not know before. And I found that part fascinating but also disturbing. So I invite you to join me on this journey with Dr. Richard Burt as he talks about the book Kill Switch, why he wrote it, what should we expect when we read it, and how much research did he do, and how did he verify all of the information that he shared with us towards the end, especially with the COVID-19 pandemic. It's really an important book. I highly recommend it. I read it cover to cover, and I strongly recommend that you also do that as well. Before I air the episode I taped with Professor Burt, I have to urge you to subscribe to my show, hit the like button, comment, join the conversation, always. And here he is, Professor Richard Burt. Well, I'm super excited to host Dr. Richard Burt today on my humble podcast. I had a chance to, I've known Richard actually for a long time, although we have not met. It's been a while since we've actually seen each other. But I was a lowly fellow when you were an attending and faculty at Northwestern University. I remember this almost 25 years ago, Richard. You haven't aged. I don't know what you're doing.
Well, I am working with iPS cells, and we do have patents where we can actually partially reverse aging in animals. And that's new technology that I hope someday to write a book on. But we're diligently working on that now to try to get it from animals to humans.
Oh, I love that. I love that. Okay, but the reason we are talking today is because we are going to talk about your book, Kill Switch, and we'll get to it a little bit in a little bit. But before we do that, I want to make sure listeners and viewers get to know a little bit about you and how your career and what you do. And then from there, what got you to decide to write this book?
Yeah. So I was a fellow at the NIH in the last century, and I had this idea of using stem cells for autoimmune disease, and it was just kind of this fire in my stomach, a passion. And so I was, as part of my fellowship at the NIH, they farmed me out as a fellow at Johns Hopkins for stem cell transplant for oncology because they weren't doing that at the NIH. And I really enjoyed that. And some of the professors, I talked to them about this concept, and they were very supportive, especially Professor Bill Burns. So then I, started working in his lab, and I went and spoke with Art Nienhuis, who was the head of the hematology branch, and said, I wanted to work up at Hopkins under NIH salary. And he kind of leaned back and looked at me and then said, okay, as long as you do your clinic here. And so that worked out well for Hopkins. They got a free fellow. It worked out well for me because I was doing research on what I wanted to do. And Bill Burns emphasized that we have to begin with an animal model. And so we began with an animal model of MS called EAE and started working there. And so long and short, I spent 10 years working on animal models and eventually learned a lot from them and how applicable they are to human disease. And so then I went around talking with people about starting transplant for autoimmune disease, and Northwestern agreed to let me do that. So I went there, and I was the head of allogeneic transplant for malignancies, but I continued working on animal models for autoimmune disease and put in trials. And I got a big NIH NIH grant for around $10 million. And with that, I was given my own division and I gave up cancer entirely and just focused on autoimmune disease. And that allowed me to really become the world leader in that. We did a lot of stem cell transplant, highly efficacious. We did the first randomized trial for multiple sclerosis. We did it for scleroderma also and a variety of other diseases. But then along the way, I had another idea, about using stem cells, embryonic or iPS cells, for regeneration of tissue, especially for aging or traumatic injury. And we started working on that, got 7 patents on that, and then eventually got the university license back to me and separated from the university, retired, and continued that work, which we're trying to bring to fruition. But while all that was going on back in 2019, one of my fellows came to me, and he was from China, and he said, this Dr. Shi in Wuhan, he was following her. She was very famous in China because she'd gotten NIH grants in China at her institute, the Wuhan Institute, to do a bunch of virology or looking at different viruses, especially the COVID virus. And she had the nickname in China, the Batwoman. And I'd spoken several times in China as well. I had never met her or been at that institute, but my fellow said something was wrong. Everything on our website is just totally cleaned in one day. It was there, the next day it's just gone. And so I began to look into that. And COVID the story was coming out in America. And as I found that, you could find the NIH grants, because I had NIH grants, especially one big one. And you can find them on websites. And so I cross-referenced her to NIH grants and found out that there was a big NIH grant that had gone through EcoHealth in New York from NIAID to Wuhan Institute to Dr. Shi to continue genetic manipulation of the COVID virus. And the COVID-19 is a COVID virus. And so that kind of got me interested. And because I had been on media for my publications in transplant for autoimmune disease, I contacted a broadcaster on a major national, international corporate media group who I knew. And I said, look, the NIH was funding this exact same research. Here's their grant to develop a COVID virus that would infect humans. They were doing that. And I said, people need to look into this because now we have this COVID problem. And, he sent it up the chain to a producer, and a producer wrote back very nicely, well, these things are really complicated, and if we have questions, we'll contact you. They never contacted me, but I read the grant, and so I knew something wasn't right. And I guess in fairness to them, they're not molecular biologists, they're not researchers. They probably would have some difficulty reading it and understanding it, but it was clearly what they were doing was manipulating the binding of the COVID virus to human epithelial cells to maximize its potential to bind to cells to cause disease. And there was such media hype, and anybody that mentioned it was attacked and considered a conspiracy theorist, that I just didn't follow up with anybody, continued doing my work. But then I stumbled across another grant by this same group, Chapel Hill and EcoHealth in New York and Wuhan, China, for development of the furin cleavage site for this COVID virus. And the Department of Defense refused to fund it, but I read that grant as well. And again, doing grants, you know how to write them and you know how to read them. And I've sat on NIH grant proposal committees and things. So I was like, whoa, this second grant from that same group said that they were going to insert the furin cleavage site in a COVID virus. And that site is exactly what the site is in COVID-19. So to take a COVID virus that doesn't infect humans and make it infect humans, you have to change the receptor binding domain so it'll bind human epithelial cells. And they did that, and they reported in the scientific medical literature that they were successful in doing that. And what that'll do, it'll allow the virus to infect cells, cause infection and an epidemic, but not a pandemic, because for a respiratory virus to go from infecting a small number of people to becoming a worldwide pandemic, it has to have another change in the virus that allows it to be easily transmitted between humans, not just infect human cells, which can cause infection in a relatively limited number of people before it fizzles out, but to be easily transmitted. And the furin cleavage site does that, because what that does is it changes another part of the binding receptor, another domain, so that when that receptor binds to human cells incorporated inside the cell, furin, a protein that we carry in all our cells will cut that binding site and open the virus up. When it opens up, it allows the virus genetic material to be easily replicated and rapidly replicated instead of the slow opening that naturally occurs. So it enhances transmissibility. So that's the second step needed to make a pandemic. And that's the exact molecular signature of COVID-19. So I realized, this what most likely had happened. And I just felt there was so much misinformation out there, although it's a little bit of a detour from my traditional and current focus. I felt it was important for humanity to write Kill Switch to get the information out there, not because I wanted to criticize anybody. That's not what I do if you read it. I don't do that. I certainly don't attack people's motives. I believe everybody had good intentions, but to show how good intentions can go bad and to show how the field of virology has changed from developing vaccines for known pathogenic viruses to now creating new living pathogenic viruses, pathogenic to human beings that have never existed on this planet. And that can result in disasters. And although there isn't proof and you have to draw your own conclusions, the preponderance of evidence currently suggests it was a result of NIH funding via EcoHealth in New York, the Wuhan lab, to do exactly that. And that's what I bring out in the book. But I have to bring out the historical perspective to kind of point out how we got there, because, if you just hear the story, you think, well, that's crazy. Why would anybody do that? But when you go back historically through viruses and their impact on human beings, and actually a vaccine is a kill switch to a pandemic. But once you start creating new pathogenic viruses that have never existed on this planet, if you're not taking the precautions for the appropriate oversight or the appropriate kill switches built into these new viruses, you're risking a worldwide pandemic. And I wanted people to be aware of it so it doesn't happen again in the future.
But what's so interesting about what you did is you actually do go back to talk about other viruses. In other words, the book doesn't necessarily talk only about COVID. In fact, COVID, there's only one chapter on it. It's a long chapter, but there's only one. And you start by talking about smallpox, and you talked
Yes.
about so you went back to go through all of the viruses. So before I go through this, though, tell me the title. Tell me a bit about help listeners and viewers understand the title. How did you come up with the title? Because when you think of Kill Switch, I don't necessarily think you're going to talk about viruses. It could be like anything. Like I wasn't really sure how the title came about, and I think you do thank Senator Mark Kirk, I believe, for suggesting the title and so on. Take me through the story that led to the title of the book.
Well, Senator Kirk from Illinois has become a good friend of mine. We do get dinner together, and I was having dinner. And when all this was going on with COVID, I explained to him what happened. And, I said I might write a book on this. And he said, good, the title should be Kill Switch. And so I do give him that credit in there. But a kill switch is really anything that stops good intentions from going bad. It's being able to put the brakes on something when it goes bad. Like brakes are a kill switch on a car. There are traditional kill switches that are very important. Outside investigation is a very important kill switch. So we as individuals, if accused of a crime, are not allowed to investigate ourselves. But government institutions, whether it's county, city, state, or federal, routinely investigate themselves. And that's not a good kill switch. You need outside investigation. And that's actually what happened here. The NIH that was funding this investigated itself. There's an inherent bias or conflict of interest. And so that shouldn't be allowed. It's like, and I don't mean to stray this in a different way because, there are many good police out there, but there are times when things happen to citizens and you have police officers' word they did bad. And then when you look at the videos that were taken and you realize the police were wrong. So those videos are a kill switch. But when it's investigated within its own departments, it's often said, oh, there's no problem here. Nothing was done wrong. Only when the videos are released to the public is there an uproar, and then there's an outside and appropriate investigation and accountability. So outside investigation is a very important thing, and that's deficient when we're dealing with the government. And hopefully we need to realize that and change the way we have investigations occur with city, state, county, or federal governments, their institutions, and their departments. A classic example, again, is all the confusion and misdirection that happened under COVID-19 because it was an inside investigation from the institution funding it. It wasn't from the outside. That's traditionally important kill switch. Another important kill switch is freedom of speech. And there was this, well, you can't ask questions or, something's wrong with you. No, you need to be able to ask questions. That's what science is. Science is never, ever for certain. Science, as I quote Richard Feynman, is a culture of doubt. Anybody that thinks science is certain is just wrong. That's what Richard Feynman said. And that's important. That's what I know too. It's a culture of doubt. So you've got to be able to ask questions. There has to be that freedom of speech to ask those questions without fear of retribution or having a big sign hung around your neck of adulterer and exclusion, because it's important.
why did you want to write about the other viruses though? Like, was your goal of
Well,
writing about
because
them? Yeah.
I think what happens is you have to put it in the entire context of human history to understand how we got there. If you just write about COVID-19, people will think, well, you're
I think people
against
made up their
research
minds.
or
Richard,
yeah,
don't you think like for COVID-19, most people made up their minds, whether whatever it
right. And,
is.
and and that's excellent way of putting it. The best way of changing people's minds is go back to the beginning and build it up as a logical explanation of how we got here and what happened. So that's where I felt it was the best way to do it. It also helped remove some of the emotional impact out of it to get people to think and to question. So the whole goal of that was again that the kill switch is a check and balance. The book itself is to help hopefully bring about some changes in our laws and our institutions so this doesn't happen again. And then finally, kill switch in this case has to do not with these traditional constructs of outside investigation and freedom of speech, but also with if we're going to make new viruses that have never existed before on this planet to infect humans, we should consider kill switches, technology inserted into those constructs that we can shut them off if they go bad. And, that's what I bring out in the book that has been missed to date. So, and if people don't learn from the past, it's going to repeat itself. And it won't be 8 million or 10 million dying. It will be a heck of a lot more dying. And that's why I think it's very important we take a look at that. And, it's a new technology and the regulations and the science behind it are lagging. It's kind of like developing AI. The control or regulations or concerns about that are lagging behind the technology. And so I wanted people to understand viruses and molecular biology that are lay people without a medical or scientific background. So I wrote it for people without that background. And I wrote it in a historical perspective because I love history and history makes it interesting.
Yeah.
And it builds up then so people can understand how we got here and why we need to make some changes.
And those are two important points. One is the book is not written for the scientist. This is written for the general public. Would you agree? I mean, this is written for anybody that is curious, but you do not need to have a medical background or scientific background to enjoy the book because you put on so much history. I mean, I felt I was reading a story. You're an excellent storyteller because let's be honest. I mean, when you tell somebody I'm gonna like pick up a book about viruses, it's not like, the most appealing thing that somebody's gonna think about. However, I want to make sure that listeners, viewers realize that this is not a textbook. This is really stories about viruses, about scientists. And I mean, just one example of this I put here. I mean, when you talk about smallpox, you talk about the last documented case was in Somalia and so on. But then, you talk about how we got to the extinction of smallpox, and you literally take us through the process that took that. So, how much research did you spend to write this? I mean, how many was it? Like a couple of years to be able to get there? Like, there's so much information here.
Well, yeah, so that's a good question. Part of it is I love reading and learning, and part of it goes way back when I was a fellow at the NIH. I had gone there after being a chief resident at Baylor. I was a very good clinician. I thought I was really good. And my attending from NIH thought I was good. And so they encouraged me to apply for the fellowship, and I went there. And all of a sudden, went from being what I thought was brilliant to a student again, because I didn't know molecular biology. But after spending 7 years at the NIH in a lab in molecular biology, I understood it and became good at it. And so even though I'm not a virologist per se, I recognized that there was a problem here and that people need to be informed as to what really happened. And I felt that because one of my skills is being able to explain more complicated things in a simpler way. That was my goal in writing that book, but also by putting it in a historical perspective, which I love history. It makes it interesting for people.
It
They get
was for sure.
riveted to the story. So, because you're right, if you read a science paper on viruses, unless it's really your field, you can be
Right.
put to sleep pretty fast and like, what are they talking about? So everything in there is written in a way to make it
I mean,
understood by people that aren't experts. And
let
so I'm
me
glad
let
that,
me tease. I want to tease listeners and viewers on this. Like, what one thing you talk when you talk about smallpox. I mean, literally, you say that smallpox caused the collapse of the Aztec and the Inca empires.
true.
So tease us a little bit about this. I mean, this is a virus that led to the collapse of empires. I mean, this is like so much history there, and maybe just a little bit on this. Like, how did this actually happen?
Yeah, well, the Europeans brought smallpox to the New World. The conquistadors did. And so it really wasn't their sword that conquered the Aztec or the Incas. It was that small, invisible virus, smallpox, that did it. Because being a new virus, they had no immunity to it. And, it killed well over 20 million native
Right.
Indians. The exact number is unknown, but well over that. That was much more devastating to them, to their ability to organize, to have a civilization or to fight, than the muskets of the conquistadors. And of course, it was unintentional. At the time, the Europeans probably didn't even recognize that they were bringing that virus with them and infecting them. Although I do bring out a point in there during the American Indian Wars, the Indians were given blankets laden with smallpox virus, to try to quell and terminate the Indian warriors, and that was done intentionally.
Amazing. And then every time I like was reading about something like, when I read about the yellow fever, I got to learn where the term quarantine came from. Like
Yes.
I didn't even know where it came from. I just knew quarantine, but all of a sudden you drop something and like I highlighted this. I love that stuff. I mean, tell us about this quarantine. I mean, people don't even know where the origin of that word came from.
Yeah. So again, that goes back to knowledge for knowledge sakes, which I agree with. And so I understand why you want to create these new deadly viruses. You gain a lot of knowledge, and the benefit from that comes out a lot later. So the understanding of words, I got a lot out of when I was in high school taking Latin. And because a lot of medical words, a lot of legal words, and a lot of everyday words arise from a Latin root. And so the word quarantine comes from Latin. And I bring out throughout this how many of these words, what they really mean in terms of Latin or Greek, so that we understand, because we've kind of lost the meaning. We flip these words around, but lost it. But quarantine means 40 days. And so it's isolating an infected person or group of people for 40 days, if, so the infection will pass or they'll die and it'll be gone and they don't infect someone else. And where that number comes from, nobody really knows, except perhaps an explanation is that, in the Bible, Jesus resisted the temptation of the devil for 40 days in the desert. So maybe that's where it came from. But that is an excessive period of isolation for most viruses, as I bring out in there.
Fascinating. So I mean, I want to obviously talk about the other ones, but I have to say we have to spend a little bit of time talking about what you wrote about polio and what you wrote about HIV, because they're really, really, there's so much information there, and I'm gonna have to assume that a lot of people don't know this information because we just take things for granted. One of the things in the polio, as you describe polio, and there's again, for listeners and viewers, please check it out. There's a lot of history there. But you talk about vaccine-induced paralysis, and you talk about, which to me, again, I think you're highlighting that the vaccine is intended clearly for good reason, but there has been vaccine-induced paralysis with the polio that you wrote about this, and you talk about the last polio infection was in 1979, but there were other cases of paralysis happened after the vaccine. So can you help help us understand this a little bit? Is this, aside from information, were you trying to convey to the reader that vaccines could cause problems and don't really assume that vaccines are never without issues? Like what was aside from the information and the history, I felt you wanted to at least convey to us that these vaccines could have problems, and we have an example of that as early as the polio days.
Sure. So, as a physician, every medication has side effects and can cause problems. And one of the things young doctors don't do when a patient comes and sees them is consider that their side effects or their complaints or their symptoms are iatrogenic from medications. And I have to repeatedly stress that because older physicians have come to understand that and know that. So for every intervention, every medication has a good and a bad. It's the same with radiation. It's used to treat cancer, but it causes cancer. So it's all in the context which you're using it. And there are side effects that you have to be aware of that are important. You have to determine risk benefit. Well, for the polio vaccine, the Salk vaccine, was a dead virus. It was still quite capable, even though it was dead. It couldn't grow, it couldn't divide, but its three-dimensional structure still existed. So your immune system could develop antibodies to it, and you could become immune to polio. And the Salk vaccine was the first vaccine out there, and it did succeed. There was a little bit of a fiasco with the Cutter incidents where they didn't adequately kill the virus, and that caused some deaths from paralysis. But if the virus was adequately killed, it didn't cause any illness, and it generated immunity towards the polio virus. The problem is, you have to be injected like 3 times for this to work. And that was Salk. Then Sabin had emphasized the use of an attenuated live vaccine, not dead, but live but attenuated. So attenuation means you pass it in cells, really monkey kidney cells, repeatedly until it becomes weaker. And when you inject it, it doesn't cause paralysis, but you get immunity to it. And because a live virus is more immunogenic than a dead one, you would only have to take the vaccine once. And the advantage of the live polio is you could just drink it. So it's a lot easier to administer and a lot cheaper. So people switched to the Sabin and stopped giving the Salk virus. But the problem with the Sabin, it's a live virus. It infects people. It can exist in the gut, the small bowel, the epithelium, the lymphoid patches or pyre patches of the small bowel for decades. And that you can excrete that virus, but it can revert back to the pathogenic state. And so now by far and away, the most common cause of the actual only cause of poliomyelitis paralysis in America is from the Sabin vaccine. And because of that, in America, they switched back to Salk so that we won't be having a Sabin vaccine-induced paralysis. But Sabin vaccine, because it's so inexpensive, is still what's used and easy to administer. It's what's used in India and Africa and other parts of the world for financial reasons. But there's something we have to consider the ethics of what we're doing here, because, if for America we say it's not safe and we want to use the slightly more expensive Salk so we don't get vaccine-induced paralysis from revision back to the wild type of pathogenic polio virus, it's like, why don't we give those same standards to the rest of the world? Not only because that's the right thing to do for them, even though it might be a little more expensive, but also because of air travel. Now, people travel everywhere, and if someone gets it in a different part of the world, they can bring it back to America. So, it doesn't quite make sense. And so I bring out viruses are wrapped with a lot of ethical considerations and conundrums that need to be considered while the field's developed. And probably one of the best way to deal with them is be absolutely honest about the good and the bad of viruses and vaccines. And so I even begin in the integral part about how viruses are maybe the foundation of life and an integral part of life, an integral part of who we are, an integral part of our genome. But of course, viruses can cause a large number of deaths, and that's how we end up developing vaccines. And then I get into some of the problems of some of the technology technologies used to develop these vaccines. Not because I'm saying we shouldn't use a vaccine, but that we can improve it and we can make it better, and we shouldn't ignore problems. Problems are our teacher.
What are the chances of developing polio vaccine induced paralysis?
They're small. There's only about, 1,000. If I'm going to have to double check this, but maybe 1,000 cases a year in America. But yeah, all those cases have been related to the Sabin or oral polio vaccine.
You moved on then from polio to HIV and AIDS, and I think a lot of people probably will relate to that more because I think most, again, this happened in the 80s, so a lot of folks who dealt with COVID-19, obviously, probably there are some more vivid memories of this of the HIV problems that occurred. I want to talk about a couple of things. First, you describe several types of the HIV, and you describe several stories at how humans contracted HIV. Do you want to tell us just briefly, just give us the story of how humans got the HIV and the different kinds of HIV? And then I
So
have another
there,
question about that.
Yeah, there are different types of HIV, but the one that became a pandemic went worldwide is HIV-1 group M. So that's the only one really to talk about. And the question that comes up is, where did that arise? And the canonical answer, the accepted classical answer, is that this HIV virus is very, very similar to an SIV virus in primates, and so the concern is that humans ate bush meat of primates, and that's how we infected ourselves. But there's another hypothesis out there, and that hypothesis was first written by an investigative reporter for the BBC named Edward Hooper, and what he showed is that HIV originated in Kinshasa, the Congo, and in the larger cities in the Congo, not in the countryside where people would eat primate bushmeat, monkeys and apes and things like that, but in the cities where that wasn't happening. And that occurred at the same time that they were making a vaccine for polio. And so the problem is the vaccine is made in monkey kidney cells, and monkeys are the natural reservoir and source for human HIV. In the monkey, it's called SIV, simian immune virus. And it's the chimpanzee is the direct, not the regular monkey, but the chimpanzee is the direct source for HIV that spread worldwide. And so, the Wistar Institute that had taken the development of the vaccine to the Congo, they had developed the vaccine in Philadelphia using brought the kidneys of monkeys who were bought and brought over from India. But those monkeys don't have SIV. They don't. And so their kidneys, when they would culture the kidney cells and grow the kidney cells to use those cells to grow the polio virus, there was no risk of them transmitting HIV with the vaccine. But the question is that what happened in Africa? And nobody's ever investigated that. Hooper's book suggests that is what happened by epidemiologically correlating the two together. But the answer to this day is still unknown. What is known is that chimpanzees are prevalent in the Congo, that in the Congo at that time, even up in 1976, 1980, a nurse would be given like 5 needles and syringes, and they had to use them over and over and over because they didn't have the money to buy a fresh needle or a fresh syringe for a patient, and they wouldn't sterilize them. They'd just wash them out in between. Well, that would allow transmission of viruses, hepatitis, HIV between patients. But if a country didn't have the money to buy fresh needles and syringes like America was doing each time on a patient, why would they buy monkeys in India, pay for them and have them flown, pay for that expense to bring them to Africa when they could use chimpanzees that have bigger kidneys and you have more cells to be able to produce more virus? So it would make sense because nobody knew there was a risk that they would have used chimpanzee kidney cells to grow the polio virus to make the vaccine. But the problem is those chimpanzees in Africa are contaminated with HIV, and they could have then passed HIV on with the vaccine. Did that really happen? there's never been an adequate investigation to understand if it did or not. There has been a semi-attempt with phylogenetic studies where they tried to use the rate of change in the nucleotide sequence of the virus to predict when it first entered the human population. And those phylogenetic studies, they were very well done. They were reported in Nature and in Science. They were out of Los Alamos here in America. I spoke with the lead investigator in the study, but those are based on two things. They're based on the number of samples you have, the size of the samples. And there's no sample available before 1959. And we know in 1959, which was 2 years after they started using the polio vaccine in the Congo, that virus was found in a patient that they were doing another study on for a different reason, and it saved the serum. They went back and looked at it and found HIV in that patient in Kinshasa. That's the earliest known case. There's nothing before that, even to this day, because there's no saved blood samples from before that. But when they would do the projection back based on a constant rate of nucleotide change, it would fall back into the 1930s or 1940s, well before the polio vaccine, which began in 1955 in the Congo. But the problem with that, it's based on the assumption that the rate of those changes are constant, and that's not true for HIV. The mutation rate depends on the rate at which that virus proliferates, and that changes markedly based on another existing infection, and based on the fact that normally HIV, once you get infected, will integrate into your DNA and sit quiet for years or decades before it's activated by an infection. So I think we have to take those results with a caveat and say we don't yet know for sure that the phylogenetic data accurately predicts the time of onset or transfer of the HIV virus into humans. And again,
Is that why we couldn't ever develop a vaccine against HIV, or that there's another reason why we couldn't have a vaccine?
well, I think there's a variety of reasons. One of the predominant ones is that it is an RNA virus and it mutates so fast that no vaccine has been effective because of that fast mutation rate. So what happens is the virus infects a person, it gets in their DNA and it sits there, and for years or perhaps even decades, you're asymptomatic, and then something makes it break out. And what it is an infection. Will activate the virus to be transcribed from the DNA back to RNA and become infectious particles, which then infect your lymphocytes, and it decreases your immune cells, which increases the infections, which increases the virus turnover, which increases the amount of virus in the blood, which increases the ability to infect other people. So it starts a cycle that basically is activated by infections, but it lowers your immune system to cause more infections, to cause more activation of the virus, to then consume the host. But because you have such a high virus titer within your own body, you can infect other people and spread the virus. So HIV kind of found this niche, but it definitely occurred suddenly in the 1900s. Exactly when it did, we don't know. If you believe the phylogenetic study, it was before the poliovirus vaccine from the Wistar Institute. But if you look at the fact that it is highly likely, unproven and emotional, but highly likely that they used common sense and took the readily available cheap chimpanzee kidney cells to create the virus for the vaccine, they likely transferred, when they were doing that, HIV with the vaccine to people. Now that's all theoretical. Nobody knows for
Sure,
sure.
sure.
And but I think people just need to be aware of it. And one of the issues that arises is the only way to answer that is to go into the people that were doing it and ask them
Sure.
and look at the transfer of agents and look at the lab books and everything like that to do a real investigation. That was never done. And now, since that was back in the 1950s, most of those people are probably gone. But actually, Ed Hooper actually says he's talked to people who worked in the lab in Kinshasa. They told him they used monkey kidney cells and monkey serum, not monkey, chimpanzee kidney cells and chimpanzee serum in the production of the polio vaccine, which obviously would transfer the HIV virus. But it's never been proven,
Yeah.
and there seems to be no interest in looking into it. But what this teaches us is that when we use animal cells in a human being, we can get transfer of animal virus into the human population that have never been in humans before, and it can be disastrous. And actually, people understand that now. So the FDA really is very hesitant to let you use any material that has xenogenic, that is not non-human exposure in the development of the product. Now that can become ridiculous at times, but that's why that caveat exists. It's something that was learned really initially with the polio virus, even though it's not really spoken about.
One of the most disturbing things I read in the book, the most probably the most disturbing thing was the Uganda HIV study, which was published in the New England Journal of Medicine. I was reading this and I couldn't believe I was reading this. But how did this happen?
Well, when I read it, I was shocked. I was literally shocked. And I was shocked also that nobody else seemed to notice, and it's never caught attention
The
of the
first
media.
time I hear about it's I
Yeah.
literally this is the first time I hear about this study when reading your book.
Yeah, and it's a repeat of the Tuskegee syphilis study. It
Right.
is an exact repeat of it.
Right.
So I just think we need to make people aware. And again, what it goes back to is consent. Consent is an important kill switch. You must have transparent, open, honest consent. That protects patients, but it protects the investigators. And it protects society as well. So if somebody had told those villagers in Africa, look, one of the two of you of this husband-wife team has HIV, we're not going to tell you who, and we have effective therapy to treat it, but we're not going to treat it. We're just going to watch to see when the other person gets HIV, a lethal disease. And afterwards, we won't treat you and we'll say goodbye. Well, nobody's going to agree to that. So they didn't tell them the real reason they were doing the study
Right.
because nobody would agree to it. And they didn't do it in America because it would have been unethical. So, but they went ahead and took advantage of these poor villagers in Uganda. And I bring out in the book, isn't that what we do with animals? Take advantage of their circumstances. But it was a repeat of the syphilis Tuskegee study.
It gets
And for
into the
some,
NEJM,
yes,
the
yes,
most prestigious journal in the world.
yes, yes. And it was done blatantly and it just kind of oblivious to what they're doing.
Yeah.
So again, that's another important point besides outside investigation is consent. Consent is a very important kill switch for misbehavior. And so, and to get consent, you have to be honest and open with people. And that didn't happen in the Uganda HIV study. And I think people listening to us will kind of like, what are you talking about? I say, just read Kill Switch.
Yeah, no, I mean this was really very disturbing to read, and like I said, I did not really know about the story. Before I want to talk about obviously COVID, we started with COVID. We want to talk about COVID as well before we finish. But before we go there, Richard, you spent some time in the book explaining the value of mercury and aluminum as part of the vaccine. And the reason I think this is relevant, obviously, is because today we hear a lot about this, and there's the assertion by some fraction in the healthcare system or in outside the healthcare system that this is what leads to the adverse events associated with vaccines, including the autism and other things. And again, you in the book, I couldn't tell where you stand, which is an art because I think you're providing facts and you're just educating people. But help us understand this particular element.
Well, so first of
You
all,
talk about it. You talk about it in the
yeah.
book, and I think it's really important to explain to listeners and viewers.
Yeah, thank you. So again, it goes back to just knowledge for knowledge sakes because my original degree in college was in chemistry. And because of that, I was always interested in these elements. And in fact, when I added those two chapters, one on mercury, one on aluminum, I thought, gosh, I enjoyed writing them so much that I should write about every element and just do a book on it. And, there's no time for that. But I really enjoyed writing about them, giving the history of those two elements, as well as before that, a chapter on atoms, which so you kind of get an understanding of that for the rest of the book. But basically, mercury was added to vaccines as a preservative. Now, preservative is a kind of a euphemism. What's a preservative? What it really is a germicide. What's a germicide? It's something that kills bacteria and fungi. And mercury is very, very good at that. It kills bacteria and fungi, so it sterilizes the vaccine. And what basically mercury is toxic to any carbon-based life form. It is. And in its charged form. In its uncharged form, it's not. But in its charged form, it is. And so what had happened is before the addition of mercury to vaccines, because they were multi-vials, so they would put syringe in several times to draw back to give a vaccine to different people. So one vial could vaccinate 20 people. It was very cost effective. But by putting the needles in and out of the vaccine vial, they got contaminated with bacteria, and some patients died from that. So they had to do something to stop that. Well, one of the things to do is have one vial per patient. Don't have these multi-vial vaccines so that once you draw it up, you don't ever access it again and you won't have these infections. But that's not cost effective. So in terms of shipping, if one vial can take care of 20 people, it's a lot less cost in shipping. In terms of production and storage and using it, it's much cheaper. So it's cost effective. So they wanted to keep it because it's cost effective. Well, Lilly realized that mercury in the form of thimerosal, you could add it to a vaccine, and that would prevent infections and make it sterile. And so that became the standard. Everybody was doing it. But there are risks with mercury, and it depends on the form of mercury. So mercury comes in many forms. If you're dealing with just regular ground mercury, cinnabar, it's really very, very harmless. If you're dealing with elemental mercury, as long as it's not atomized into a fine mist, it's pretty harmless. If you're to us to just it used to be people would even play with it in science class in their hands, and no ill effects happen. When you come to where you make an inorganic mercury chloride, it has a lot of toxicity, but it can't cross the blood-brain barrier, so it doesn't affect neurons. But it was used to treat syphilis, as I bring out in the book, until penicillin came about and we were able to get rid of it. And but you take like 2 years of mercury chloride and have all kinds of toxic side effects from it, nausea, vomiting, ulcers, all these things. But it was thought to be effective for syphilis, and that's why people used it until penicillin came about. It's kind of like chemotherapy,
Yeah.
like their
So,
chemotherapy
so
or like
are there
metals, like cisplatinum,
so
arsenic.
I mean you
So,
talked
but then once you go to the methyl formulations of mercury, those cross the blood-brain
right.
barrier and they're toxic to neurons. And that's what ethyl mercury, which is thimerosal, does. And so I talked about the different types of methylated mercuries, and like dimethyl mercury and ethyl mercury and so forth, and how dangerous they are and what problems they've caused because they kill fungi. They have been rolled onto seeds, but when instead of planting the seeds and growing the crops, hungry people would beat the seeds down to make bread or something and eat it and then die of mercury poisoning. And so, these are or have neurologic side effects that would kill them. So even as I mentioned in there's a pig, I think it was in Romania, that they fed the feed of seeds that had been rolled in with mercury on them to prevent fungus from growing. The pig ate it. The pig started stumbling, was unsteady. The family killed the pig and ate it, and then 5 of them got sick from mercury poisoning, with the younger one dying from it. So these methylated mercuries are highly toxic to the central nervous system. That's what goes into vaccines. So that's why there's always this question about it causing autism. And in fact, if you use very, very low doses of it when there's not a blood-brain barrier, of even just of non methyl mercury, of a mercury chloride at very low doses, it kills neurons and they retract their dendrites so they can't connect with other neurons. And I kind of showed a picture of that and explained that. But once you get the methylated forms across the blood-brain barrier, there's a serious risk that we could be causing retraction of those interconnections that are so important in our intelligence and our ability to communicate with people and to be interested in the environment around us. So there's that concern. Yet when you do epidemiologic studies, it doesn't pan out. It doesn't show up. But
People who are listening now would be curious. Do you, based on your research, based on your investigation, do you have any evidence that having the aluminum and the mercury is related to autism that being sometimes stated by others? Because I think it's a very polarizing topic and polarizing subject to actually bring up. But you went there in the book, so I have to
yeah,
ask.
well, we have to, because that's where the reality is and where the world is and where the confusion is. So yes, these ethyl mercury is highly neurotoxic. Everybody knows that. Does it cause autism? The epidemiologic studies, all of them say no. So that's the quantum we have. And the controversy persists because it's known to be very neurotoxic. So if that's not what's causing this increase in autism, what's doing it? There's no other explanation given. But the point is you don't have to put it in the vaccine. Once, instead of making multi-vial doses where you draw 20 shots from one vial, you make single vial doses. So you draw it, you treat the patient, you throw it away. I mean, it's done. Each person gets one vial because under GMP production guidelines, it is very sterile. How it gets contaminated is by inserting in and out a needle multiple times for these multi-vial doses. So the whole point is you don't need to do it.
That's
But if you want to do it cheaper, you add the mercury.
yeah.
But if you want to remove any of the risk of what is known to be highly neurotoxic and get rid of the controversy, you just use single vial. That's what needs to be done. That's the answer out there. Because nobody's ever looked at vaccines with mercury versus vaccines without mercury in terms of what are the long-term consequences, which would be the right study to do. The FDA has never considered mercury a drug. They never looked at what its side effects were. It was just always thrown in with the vaccines. So, that's kind of why I'm saying there is a concern here that is legitimate. Epidemiologic studies show no association, and I bring up some of the dangers of epidemiologic studies. Epidemiology is not a scientific proof by doing an experiment. What it is, it's looking at associations, and they're very careful to try to get rid of biases in looking at those associations. But what it is associations. And whenever you do associations, you can be right or wrong. It's not proof. People think epidemiology is proof. It's not. It's drawing associations. An
Well,
analogy.
it is a tough I mean, it is. There are two views on this. Obviously, I mean, I think I'm pretty sure you know colleagues who would disagree with some of the assertion that the epidemiology is not going to lead to more definitive answers in certain situations. I don't think we just talked earlier, there's no perfect science. There's never
True.
perfect.
True.
And epidemiology is one of the fields where it's not perfect, but we have sometimes to make decisions and to find answers despite its imperfection.
Right. But epidemiology is drawing associations between events. And when you find an association that is statistically significant, then what you need to do is do the research to show a cause and effect.
Sure.
That's what you have to do. It's just like so many African Americans were sent to prison because more crime is committed, relatively speaking, by African Americans. And so they would be convicted. That association has nothing to do with proof that that person caused that crime. Vice versa. A very rich person, well established in society, they'll tend not to look at that person as having caused the crime and may because statistically there's less association. But in fact, if you do the detective work, you find out they did do it, but they often walk free. So these associations that we have in our minds, which is what epidemiology is. Now, it is a lot of hard work in science and statistical analysis to try to remove biases, but it just draws associations. If the association is negative, it doesn't prove innocent. If it's positive, it doesn't prove guilt. You have to do the scientific experiments to find innocence or guilt. That's what it needs to be done.
I need to do an epidemiology podcast. In the last segment, Richard, we'll move to a very much less controversial topic, COVID-19. Much less controversial. There's no controversy there in COVID-19, as you know, but I love the quote you had on Winston Churchill. He talked, understanding the coronavirus gain of function story is, as Winston Churchill once said, a riddle wrapped in a mystery inside an enigma. I love that. It's obviously, we're not going to be able to cover everything, but needless to say, you bring a lot of shocking events. Honestly, I mean, some of these have been circulated in the media, some of them are not. Briefly, what should we get from the COVID-19? It's hard for me to go through every single detail. Obviously, people need to read the book and so on. But tell us the cliff notes of after all of this research on COVID-19.
So I want people to draw their own conclusions. I don't give an answer, but I do clearly show what happened. And you can draw your own conclusions because the devil and the angel's in the details. And in our mass media, corporate media, you get these 5, 10-second blips that totally exclude the important details. So I've put that in that chapter, and you read through it, and you'll be left to draw your own conclusions as to whether COVID-19 occurred from jumping from an animal into humans or out of a lab in Wuhan. What do you think is the most likely
Well, my
scenario?
conclusion was it came from a lab in Wuhan. I mean,
Yeah.
I think you convinced me with that. My question to you, were you
But to
afraid?
come to that conclusion, you have to read the story and understand
For
it
sure.
of the details. And so people have to read that chapter because they're never given those details.
Yes.
Because obviously, a news program can't take an hour to explain those details. They
I
lose
think
their audience.
I think that the issue, and I have a more of a personal question to you, Richard, is were you ever afraid of when you wrote the book and you wanted to tackle COVID-19 of any backlash? Although, admittedly, and I read the book cover to cover, you actually provide facts and you did not offer your own opinion. So kudos to you because it's tough not to offer your own opinion. But were you afraid people say you're an anti-vaxxer? You are.
Yes, yes.
Did
And I'm
anybody
not an anti-vaxxer.
did anybody say that to you?
No, but I'm sure, given our society and the way it's polarized and the way people jump to emotionally charged issues rather than thinking through the details and giving benefit of doubt. And, like I say, truth is not black and white. It's hidden behind the screen. I
Why
say that in
was
the last
COVID-19?
chapter.
Why did, I mean, you talk about Ebola, you talk about polio, you talk about smallpox, how about HIV. And you go through the stories, but none of these were as polarizing as COVID-19. At least from reading, you're like, there were ups and downs, there were a lot of issues, but it seems to be that COVID-19 was the one story where there are so many taboos you're not allowed to discuss. How did we get there?
Well, read the chapter and the chapter after it that kind of gives, I go into game theory. And
Oh, I
again,
love that. I love the
I
game
I
theory.
think without a doubt, you had the people funding the COVID-19 research to take a non-pathogenic virus, turn it into a virus that can kill humans by both finding the right receptor binding domain and then finding the furin cleavage site to increase transmissibility. Those people that funded that research led the charge against, and it's our NIH, which I spent 7 years at, and is a very, very valuable institute, and I learned a lot there. It's a phenomenal organization we need, but it was the NIH that led the charge to cover it up, to cover up the possibility that they caused it.
Right.
And that's where things really shift. We used to fight viruses to try to keep them from killing large numbers of people. And that's how we developed a vaccine. It's a kill switch. Along the way, mistakes happened. People died. We did bad things that caused other problems, but it was never intentional. And we learn as we go along. But what's happened now with molecular biology is we switched to creating new viruses that are alive and have never existed on this planet and that are intentionally designed to be lethal for human beings. And that's where we don't have a kill switch or a recognition to this day by our political leaders, our legal institution as to what kind of oversight and constraints do we need to put on this.
Do you
Now,
have
it
you
is true, all knowledge is valuable, and creating a new lethal virus provides a lot of information, the benefits of which may come out sometime in the future in a way that's phenomenal to help society. But we have switched in virology from developing vaccines with their potential risks and ethical questions to prevent these lethal viruses to now making the lethal viruses that have never existed. What can go wrong? COVID-19 was a red flag, but I worry society has missed that red flag. And that's why I wrote the book. I don't want to stop creating new viruses, but we have to put constraints and much better oversight and control on it and insertion of a kill switch or mechanism one way or another if it does break out of a lab, because even under the best labs and best of circumstances, bad things happen.
how has the reception of the book been?
The people who've read it have really loved it, but I'm not a big name. I'm not an actor or a politician, so people really don't go to it. And but those that have loved it have really liked it. I gave it to the head of molecular biology at Northwestern, and he's a great, great person. Just so I'll say, I'm not going to say his name, but he's super intelligent. He's super good, but he's arrogant. But I've always been like, if you're really intelligent and you're arrogant, it's okay with me. I that's great, and And so he took the book kind of with a smile. And then about a month later, I got an email from him. He said, Richard, I read your book and I didn't think I would learn anything. And I thought, that's so honest. It's just so typical him. But then he read, boy, was I wrong. I really learned a lot.
No, I
And he said, I'm having my wife read it now. And I mean, like, that's the best
I
compliment.
couldn't agree with that more, really. I
Yeah.
mean, I read it. I hope a lot of my listeners and viewers will read it as well. We'll put a link on the podcast. I think that there's so much information, so much history, lots of storytelling, lots of lessons learned. This is completely not what I expected. It was
Yeah.
way, way, way better than I thought. And so congratulations and kudos
Yeah.
for you taking the time to
Well,
write this
well,
book.
I wrote this because I want people to be aware. I want to engage everybody in a real debate to understand the risk benefits, why this is done. There's very sound reasons why vaccines have been developed and why new types of viruses are being made, but also the risks and how this is not the regulation over those risks because the technology is ahead of the regulations, as well as the money trail. And I bring that up in that last chapter, which we haven't talked about here, but that money trail is real important in influencing human behavior. And I bring that out through the book, how so many times all these great scientists and Nobel laureates did something for no money, financial benefit for them, just for society, and how that is flipped because of well-intended legislation. And I bring that out in the last chapter, and I think that really needs to be looked at. And Senator Kirk from Illinois understands that, and he's in total agreement. But the problem is once you have that money rolling in like that, it's hard to change it.
Dr. Richard Burt, thank you so much for coming on Healthcare Unfiltered. I appreciate it.
Thank you.
Folks, thank you so much for joining me. And thank you, Richard, for coming on the show. I appreciate you being my guest. Thank you for writing the book. Congratulations. And I hope the book does very well. Folks, pick up your copy anywhere you consume books. You're going to like the book. And it's probably going to raise more questions than you think it's going to raise these questions. You can always find all the information about me on shadinabhan.com. You can follow me on Instagram, Twitter, YouTube, Facebook, everywhere that you consume podcasts. And don't forget to share with your colleagues and friends about the podcast and join the conversation. And folks, before I let you go, I'm going to leave you with one of my favorite sayings by a Lebanese poet, Khalil Gibran. Out of suffering have emerged the strongest souls. The most massive characters are seared with scars. Until next time, take care.